Actually, virus are the nucleoprotein particle. Virus are the nucleoprotein particle. It means that they are made up of nucleic acid and protein. Nucleic acid and protein. Nucleoprotein. Nucleoprotein. Nucleo means nucleic acid, and this is protein. To understand the Hershey and Chase experiment, it is very important you should know about the bacteriophage virus. The structure of bacteriophage virus is like that. This is the structure of bacteriophage virus, and the outermost structure, the outermost structure is called, this structure is called capsid. The outermost structure is called capsid, and this capsid is chemically made up of protein. And in this capsid, there is a nucleic acid is found inside the capsid. Because this is a bacteriophage virus, the nucleic acid is, here in the case of bacteriophage, is DNA. So the outermost covering of this virus, this bacteriophage virus, is made up of protein, and inside the covering nucleic acid is found. That's why virus is called nucleoprotein particles. Hershey and Chase grew two culture of Escherichia coli. Escherichia coli is a bacteria which, in fact, Escherichia coli is a bacteria in which bacteriophage virus infection take place. It means that bacteriophage virus infect the Escherichia coli bacteria. Bacteriophage virus infect the Escherichia coli bacteria. So, first of all, what happened? Hershey and Chase grew two culture of Escherichia coli. First culture with sulfur 35. First culture of Escherichia coli is grew in sulfur 35. It is a radioactive isotopes. It is a radioactive isotope, is a radioactive isotope, is used to label the protein. And in second culture of Escherichia coli, he grew the Escherichia coli in phosphorus 32 to radioactive the DNA component. First of all, it is very important to understand that sulfur 35 is a radioactive isotope, and with the help of this sulfur 35 protein is labeled, and with the help of phosphorus 32 only DNA is labeled. So why proteins are labeled by sulfur 35? Because sulfur is a component of protein. In many proteins, sulfur is main component that is found inside the protein structure. That's why here we have to take sulfur 35. And you all know that about the DNA, because in DNA phosphoric acid is found. So to label the DNA we have to take phosphorus 32. This is the reason sulfur label the protein and phosphorus label the DNA component. Then what happened after that? These two culture of Escherichia coli are infected by bacteriophage virus, So the virus that is in fact the culture which having sulfur 35 and the virus that in fact the culture having phosphorus 32. This is a bacteriophage virus which is cultured inside the cultures inside the Escherichia coli into different type of Escherichia coli. So what happened after the progeny of the virus are obtained here? We will see that the protein code is labeled by sulfur 35 by this bacteriophage virus. It means that this virus, its protein code is labeled by S35, and in this virus the protein code is labeled by, the protein code cannot be labeled because it labeled the phosphorus. So the DNA is here, the DNA is labeled by phosphorus 32. Then what happened in another experiment? What he done? These two bacteriophage virus, in one bacteriophage virus is labeled by sulfur 35. His protein code is labeled is radioactively active, and in this virus the DNA is radioactively active by phosphorus 32. These two different bacteriophage virus are again infected to another Escherichia coli bacteria to obtain the result. Then what happened? I have to draw this diagram to explain this experiment. Then what happened? This is phase grown with radioactive sulfur 35. Phase grown with radioactive sulfur 35. This sulfur 35, this is T2 phase. T2 phase, it means that it is a type of bacteriophage. Bacterial cell, when infected by these virus, its DNA is gone into the cell and the protein code is outside the cell. So when what happened, when these phases are separated by the centrifugation method, what happened? The supernatant that is found here having the radioactive sulfur, radioactive supernatant, having the radioactive supernatant. In second experiment, what happened? To testing the DNA, phase grown with phosphorus 32 and centrifugation of this medium take place. Then what happened? Radioactivity is present in pellets. Pellets means it is a group of bacteria. It means that radioactivity is shown by the cell. Radioactivity is shown in cell. Bacteria is infected here and radioactivity is shown in cell. It means that when radioactivity is shown by the cell, it means that because cell is different and here supernatant is different. Supernatant having the virus particle, but the cell having the virus DNA. So the DNA is entered into the cell, and that's why this cell show the radioactivity. By this experiment of Hershey and Chase, it was clearly proved that DNA is a genetic material.